All right, so PTM alpha is phosphorylated, and that phosphorylation activates the protein. The second type of mutation is acetylation. And so acetylation is now going to, again, happen at the lysine residues within the protein. And so this was discovered, again, by Perry Pigsiever's lab early on in 2005. And what they did in this experiment is they used nicotinamide, which is an inhibitor of sirtuin-1, and sirtuin-1 is a deacetylase. So if you put into cells PGC1 alpha and then you treat them with nicotinamide, what happens is you then pull down PGC1 alpha and you look to see how much acetylation there is. What you can see is the nicotinamide, by blocking sirtuin-1, causes acetylation to increase within PGC1 alpha. Okay, so that means that nicotinamide inhibits sirtuin-1, and that increases acetylation of PGC1 alpha. All right, so when we acetylate PGC1 alpha, what happens to its activity? Well, if you look here, this is the fold repression of PGC1 alpha by nicotinamide. So nicotinamide actually represses PGC1 alpha almost 25-fold. So when the protein is highly acetylated, it becomes completely inactivated. All right, so now what did they do in their experiment? What they found is they found 13 different lysine residues within the protein. They began mutating them to arginines, because arginines are positively charged amino acids like lysine, but arginine can't be acetylated. So what they started doing is they started switching lysine for arginine. They did it with 2 residues, and already they saw that the repression by nicotinamide went down. So if you couldn't deacetylate, or if you couldn't acetylate 2 of the lysines because they were arginines, the protein wasn't repressed as much. When they mutated 5 of them to arginine, already you decreased the inhibition of PGC1 alpha by 2-fold. By the time you mutate all of the lysines to arginine, the protein is constitutively active. It doesn't matter how much nicotinamide you add, you're not going to get much repression of PGC1 alpha. So what this tells us is that PGC1 alpha is deacetylated by sirtuin-1, and the deacetylation of PGC1 alpha by sirtuin-1 is going to increase its activity. All right, so let's go through this. So acetylation is going to decrease PGC1 alpha activity. It's acetylated by GCN5 and P300 2 acetyltransferases, and it's deacetylated by sirtuin-1, this NAD-dependent deacetylase. Okay? So then how do we then take a look at this? How can we manipulate acetylation and see whether we can play with that and find changes within the muscle? So this is an experiment where what was done was animals were injected with a protein called scriptate, or a drug called scriptate. And the drug scriptate, what it does is it's a histone deacetylase inhibitor. It is an inhibitor of deacetylation, but only the histone deacetylation. Okay, so if you inhibit histone deacetylases, what happens is you get an upregulation of other deacetylases, because basically what happens when you inhibit the histone deacetylases, there's a lot of new acetylated proteins. And so what happens is that other deacetylases, like sirtuin-1, get upregulated. So when you upregulate sirtuin-1, that is going to have a lot of the effects that we saw when we had active sirtuin-1. It's going to deacetylase proteins like PGC1 alpha. Okay, so when you block histone deacetylases, as a compensatory mechanism, your cells increase the amount of sirtuin-1 that they have. When they increase the amount of sirtuin-1 that they have, sirtuin-1 can deacetylate some of those proteins that the histone deacetylases would have gotten, but it also deacetylates its better targets. So the targets that it's really good at deacetylating, like PGC1 alpha, and as we'll see, like P53. So what happens when we add scriptate, we inhibit the histone deacetylases, increase sirtuin-1 to compensate for that. And then what happens to aerobic capacity? Well, look what happens to the ability of these animals to run to fatigue. So their time to fatigue actually goes up by about 50%. Their energy expenditure goes up because they're able to burn more fat as a fuel. And what happens to their mitochondrial mass? Mitochondrial mass goes through what we saw before, a partial mitochondrial biogenesis. It mimics sirtuin-1 overexpression. So when we inhibit histone deacetylases with scriptate, sirtuin-1 goes up. Sirtuin-1 going up, that's going to deacetylate its targets really well, like PGC1 alpha and P53. It's going to also try and get some of the other targets that histone deacetylases would have hit. But by deacetylating PGC1 alpha, we've now activated that protein. And that's really what sirtuin-1 overexpression does, is it activates PGC1 alpha, and we get that mimicking effect where we almost see exactly the same thing as far as mitochondrial biogenesis that we saw when we overexpressed sirtuin-1 by itself. So by putting in this deacetylase inhibitor, histone deacetylase inhibitor, that's going to cause an increase in these non-histone deacetylases like sirtuin-1, and that's going to try and compensate for the increase in the acetylation of nuclear proteins. But what sirtuin-1 going up is going to do is it's going to really target its own targets like PGC1 alpha and P53. Okay? So in this way, scriptate is almost mimicking exercise. We're getting the endurance exercise effects of that exercise bout simply by blocking these deacetylases and increasing sirtuin-1 levels. So how about if we do exercise? Well, when we exercise, we activate sirtuin-1. And you can see that here because when we take the wild-type sedentary animals and then we run them for a period of time, and we look at the acetylation of a specific sirtuin-1 target, P53, what you can see is that the acetylation of P53 goes down after exercise. How do we know that that's because of sirtuin-1? Well, in the MKOs, what we've done is we've knocked out sirtuin-1. There's the sedentary sirtuin-1 knockout. There's the sirtuin-1 overexpressing muscle-specific knockout. And now what you can see is because you can't activate sirtuin-1, you don't decrease the acetylation of P53. So P53 is deacetylated by sirtuin-1, and that goes up when you exercise. And when you do this, what you get is you get an accumulation of P53. And that accumulated P53 which is a tumor suppressor, could very well be one of the reasons why endurance exercise is good for decreasing tumor rate. So what happens when we look at sirtuin-1 and mitochondrial biogenesis? So in this experiment, we've taken those sirtuin-1 knockout animals and we've given them a running wheel. And they run, again, 7 kilometers, 10 kilometers a night. And when you give them a running wheel, a voluntary wheel, so these are the voluntary wheel running groups. The wild type animals, they increase mitochondrial mass. You can see that they increase PGC1 alpha, they increase all of the complexes of the mitochondria. And when we look at the muscle specific knockouts, given the voluntary wheel, they also increase PGC1 alpha and increase all of the complexes of the mitochondria. Okay? So here, even though sirtuin-1 is important for deacetylating PGC1 alpha, what we see is when we knock out sirtuin-1, exercise still increases mitochondrial mass. So sirtuin-1 isn't required for mitochondrial biogenesis. The interesting thing here, though, is looking here. Here's PGC1 alpha, where we're looking to see if we immunoprecipitate PGC1 alpha, how much acetylation is there. And when you look at this, when you run a wild type animal here, the wild type animal here goes and it runs, and you look to see how much PGC1 alpha acetylation there is. PGC1 alpha acetylation goes down after exercise because it's deacetylated by sirtuin-1. Even when you knock out sirtuin-1, though, you still deacetylate PGC1 alpha. So that was super interesting because, yeah, we got mitochondrial biogenesis, but it didn't seem like we even blocked deacetylation of sirtuin-1, of PGC1 alpha. So how is this happening? What's going on in these animals? Well, interestingly, GCN5, the acetyltransferase, normally interacts with PGC1 alpha. But when you do endurance exercise, here are the sedentary animals here. This is when you've pulled down PGC1 alpha, how much GCN5 do you get? And you get a good amount of GCN5. You then run, and you then have the animals run, whether they have sirtuin-1 or not. And then 3 hours after they run, you pull down PGC1 alpha, and how much GCN5 is associated with PGC1 alpha? It goes way down. And so what we are doing in the knockouts is it doesn't even matter whether sirtuin-1 is there or not as far as the deacetylation of PGC1 alpha. Yes, when sirtuin-1 is there, we get a nice deacetylation, but when sirtuin-1 is not there, we also get a deacetylation, because when you do that exercise bout, the GCN5 no longer interacts with PGC1 alpha. So it's not necessarily that we're deacetylating by exercise, but what we're doing is we're not adding new acetyl groups in, and we are activating sirtuin-1, so we're really shifting it towards deacetylation. When we knock out sirtuin-1, we still get deacetylation because we decrease the interaction of GCN5 and PGC1 alpha. Because remember, there's two ways that you can decrease the amount of acetylation. One is by decreasing the activity of the acetyltransferase, or the other is activating the deacetylase. Endurance exercise does both of them. So it really deacetylates the PGC1 alpha protein because you really want to drive up the activity of the protein. So what it looks like is this. When you have, during exercise, you're generating NAD that's activating sirtuin-1, and that's causing deacetylation. But when you exercise, you also, and we didn't show you this, but you also activate AMP kinase, and that inhibits GCN5. GCN5 can't interact with PGC1 alpha, and that's also going to lead to deacetylation. So both of those two things together are going to end up with all of the PGC1 alpha in the deacetylated, highly active form. And then you're going to start making a shorter form that doesn't have the repressive domain, and it's still going to be regulated by deacetylation or acetylation deacetylation. But what we don't know yet is how that shorter form, the deacetylation of that shorter form is affecting its activity.